Yeah, yesterday we were talking about the replacement series approach. And we said that with the replacement series, you are using one density. At some point, initially we talked about the additive designs. And we said that you can alter the proportion when you are increasing density. And it's actually a problem when it comes to interpreting the results. The replacement series also has its problems of using one density. So there is an experiment which is thought to be an improvement of the two. This experiment is known as or study is known as the addition series or complete factorial. But sometimes because of too many treatments, incomplete factorial designs are sometimes used. It also uses a response surface for interpretation of the results. Sometimes it's just referred to as the response surface experiment. So we can literally improve the replacement series by including many densities in one experiment. Because the weakness of the replacement series is that it operates using one density. So what we just do for the addition series, we're just increasing the number of densities. Now the response surface is a kind of a graph which is used to interpret the results. The disadvantage of this experiment is that it may become very big, but in some cases, incomplete factorial designs are used if the experiment becomes too big. There's an author, Swanton et al., who actually summarized most of these studies. Later on, you will see the details about this paper. So I want to focus on the type of treatments which are used in the addition series experiment. Yeah, we are already familiar with the replacement series experiment, which employs proportions. We can use the example of Rapoko grass and the maize proportions. Okay, we can start with the Rapoko grass at 100. Maize will be 0. Then at 75 Rapoko, we have got the 25 maize plants, 50. Then we have also 50 maize. Then we can have 25 Rapoko and 75 maize. And number 5, we have 0 Rapoko and 100 maize. You can see as the Rapoko is decreasing, maize is increasing. So from treatment 1 up to 5, we have got the proportions of Rapoko grass and the maize. And we are operating at 40 as our density. That is the total. Now we can also change the density up to 60, but maintaining our proportions. And you can actually increase it up to 160, but still maintaining our proportions. Now, this type of experiment can be a 3 times 5 factorial. We have got 3 densities, right? And we have got 5 proportions. So it's a 3 times 5 factorial, which can be replicated maybe 4 times. Once we have done that, then we now have an addition series, and we would have solved the problems of the replacement series and the addition, the replacement series and the additive design. Okay, so I think at this stage, I will stop presenting this PowerPoint and move to the whiteboard to explain something. I now want to share the whiteboard. Hello.
Hello, mamuka Stanford Mabasa.
Tamuka.
Tamuka ndisimba.
Wazokwanisa kupinda here?
Itirili pediatric yachaisika na kumbobatsira na botsa ya hospital leptopo. Hapana.
Anotozokutumira ningi.
The audio.
Yes.
It's okay.
All right, thanks.
But ndinga sarira dhoku mukumbozvona.
Tatotanga ka.
It's okay.
Tinozoita in touch.
Paripo.
Okay.
Thank you.
All right. Okay, sorry about that. Right. So we want to look at the objectives of the addition series. We want to determine the competitive ability of both the crop and the wheat. Competitive ability of the crop and the weed at different densities. So number one, to determine the competitive ability of the crop and the weed at different densities. The other objective is to determine intraspecific and interspecific competition. This is the second objective. And remember, we are talking of what is known as the addition series. Don't confuse it with the additive design, the addition series experiment. So the addition series experiment has got two objectives. Number one, to determine the competitive ability of the crop and the weed at different densities. And number two, to determine intraspecific and interspecific competition. And we said it is normally a factorial experiment. Which can be complete or incomplete. So basically, it's a factorial experiment which should be complete, but it becomes too big. So in most cases, we can have incomplete experiment. This is to accommodate a high number of treatments. Now, I want to come to interpretation of results, but what is measured here is biomass. So data So data which is collected from this study is a biomass. So we want to move on to how do we interpret the results. Interpretation of results. So when we have collected our biomass, we have to draw some graphs, and we may have to use some equations. I'm going to start with a graph which you may be familiar with. So I want to draw this graph. On the x-axis, we are going to put plant density. They will represent it by D. Then on the y-axis, we need yield. So let me write yield here. Now, you may be familiar with this issue that if you increase the plant population of a crop, you increase yield up to a point where the plant population is optimum. So we get something like this. Even if we increase the plant population, the yield stabilizes. And at this point, probably we're having the maximum optimum plant population, sorry. And this is also a hyperbolic graph, a rectangular hyperbola. We may write an equation to represent this graph. Here where it starts, our slope is 0. So we can have B 0. And this slope is B1. But sometimes the graph can start at this point. And B may not be, 0. But it's starting at 0, so our B is 0. And we've got density here, which is D. So we can describe this graph by an equation which is like this. We put D here for density from here. And it is, we put a division sign there. Then we put brackets, B0 plus B1 multiplied by D, or in brackets. Let me indicate properly this bracket so that it doesn't look like a C. Okay, I've made it worse. Sorry, hello? Tamuka.
Ndachikumbira kubvunza kuti muri muofisi menyu pane varikuda kuitawo audit yeICT varikuda access kwamaisvo apa?
Uyai yenyu mutore makeys, ndiri mukati melecture.
Muri theatre ipi?
Ndiri muofisi.
Alright, okay.
Uyai yenyu mutore makey.
Alright, thank you.
Okay. Sorry about that. So I put this bracket. So when we solve this has to divide into D. So this is a hyperbolic, a rectangular hyperbola, this equation. And it is describing this graph coming. Yeah, tarisai yeah. Write it down. Hello, how are you? Yeah.
Hello.
Yeah, it's all right.
Thank you.
Okay. Sorry about the disturbance. So if we increase density, this is how yield is described by this graph. And this equation is representing this graph. Now we want to look at a situation where we have got one plant. Here we are looking at the total plants. If we are measuring the biomass of the total plants or their yield, we get this shape. So I draw another graph. Again, we should have a plant population down there. Plant density, sorry. Then here we have got individual plant. We can represent it by W. Remember, when we were dealing with the first graph, when we increase plant density, our yield was going like this. But when we deal with one plant, we get this sort of a curve instead of this curve. So it has to be like this. So again, this is a rectangular hyperbola. If we were considering all the plants in the population, then we would produce something like this, or this graph. So this is the yield is for all the plants in the population. This is the yield of an individual plant. That's why we're having this sort of curve. We can make this a straight line like this. If we plot 1 over the individual weight, we can get a straight line. And this is what we are interested in. Again, I write plant density here. Here I write the 1 over to show a reciprocal. So I'm dividing the individual weight into 1, which is a reciprocal. This is D, our density. Now this becomes a straight line. So instead of having B0 down there, B0 is now here. And this one is our B1. Now, if we can now write an equation which describes this straight line like this. 1 over W equals to B0 plus let us call this BC to represent the crop, multiplied by D. This is the equation of a straight line, which you may be familiar with. So this is very important because this model could now be used to assess with competition in the addition series experiment. So we are mostly interested in this. Although we started from here, because we need this graph to explain what is happening here. And also, we have now transformed this graph into a straight line. We have converted it into a straight line. And it is this equation which is important. Now, if I move a step further, we want to look at a crop. Maybe I use a stick. Crop in a pew stand, or in monoculture, sorry. You can use the term monoculture. We can describe this by an equation which is 1 over W equals to BO plus BC for the crop, D for the density. And for the mixture, now for the mixture, we're having a crop and a wheat. So we wanted to have a look at a situation where we're having the crop and the wheat. Now we can write this expression to describe the situation in a monoculture. Let us now describe the situation in a mixture where we have got the wheat and the crop. We can use, okay, let us put this C here to represent the crop. Coming. So we have got BCO, right, plus BCC. Then we have a D and a small c here. Let me correct this. Sorry about this. So that is a C plus this is B. Then we have got C. Okay, let me correct it. C W, then D W. Right. I wanted to explain this. This BCO here is what they call the intercept. That is where the straight line begins. Then the BCC is the other slope. Of the crop. This is the density of the crop. Now, this one is the slope of the wheat, BCW. We have used those small letters as a label for the slope of the wheat. And this is the density of the wheat in the mixture, DW, and its corresponding slope. This is the density of the crop with its corresponding slope. Now, let me write this. Okay. This represents the crop. I must write somewhere crop. Let me write here. Perfect. Crop. Now I want to write the expression for the wheat in the mixture. It will be 1 over W, small w representing the wheat. Then here we have got a B WO representing the intercept for the graph with respect to the wheat. Then the slope becomes B. We represent it by 2 W's. Then a D plus another W representing the wheat. Then we write this slope for the crop. Here we had CW. This now becomes W. C. Can you see that we have swapped? Here, because it's a crop, we have to start with the C, then we put W. Here, it's a weed. W, then C. The density for the crop, we put a C here. So this is the expression for the wheat in the mixture. We start with this, what we call the intercept. Sorry, let me wrap this. This is the intercept. Now this is the slope with respect to the density of the wheat. This is the slope with respect to the density of the crop. Now, I want to highlight these small letters which we are using, what they represent in this equation. But probably I should write the name of the scientist who proposed these equations to describe what is happening in a mixture before I proceed. This was proposed by Peters in 1983. So he described a situation in a monoculture using this simple equation for the crop. Now, if the crop is now infested with the weeds, we can describe this is the weight of one plant of the crop. We can express its weight using this expression. This is the weight of one wheat. We can actually express this weight using this equation. Now, what is important with these equations? Let me use a different color, like this one, or blue. I want to underline BCC. And BCW. Now, these two slopes can actually give us what is happening in the, it can tell us about the type of competition which is taking place within the mixture. I'll also underline this for the wheat. Let me use a brown color. Now, BCC defines intra-specific competition. And BCW defines intra-specific competition. So here we've got BCC showing us the intra-specific competition in the crop. Here, BWW is showing us intra-specific competition within the wheat. So with this experiment, we can now measure, or we can now estimate competition. This CW represents intra-specific competition. And also this one is representing intra-specific competition. Probably let me just label it here. So it is this and that. Then inter-specific competition. It's this and that. So we can use these are just slopes of graphs. So we can use these slopes or these coefficients rather to measure intraspecific competition. So this is how we arrive at intraspecific competition. The other thing which we can measure is what is known as the competitive ability. Let's say we want to calculate the competitive ability of the crop. We divide this value here into this one. Then we can get the competitive ability of the crop. If we divide this BWC into BWW, we get the competitive ability of the wheat. So let me get ahead and write competitive ability of the crop. This one is equal to B C C divided by B C W the competitive ability of the wheat is equal to BWW over B. Here we just swap, it becomes WC. So these values which we get for the wheat and for the crop, it can tell us which is more competitive. We just look at the magnitude of the size of the numbers, so that we can rank them or check which is more competitive. Right? But these, we know they can tell us about intra-specific competition. In a crop, this one in a wheat, then intra-specific competition, this expression can give us intra-specific competition as well as this one. Then there's another term which is known as niche differentiation. And before I move further, we are getting these slopes to compute further the other terms, competitive ability of the crop and the competitive ability of the wheat. So there's another term which is known as niche differentiation. Index. Or in bracket NDI. Now I will explain its importance, but now I want to just show you how we calculate it. This one is equal to let me move back. We take this expression for the crop. It's okay. It's B C C divided by B C W, or in brackets multiplied by, or in brackets B W W divided by B W C. So this is what is known as the niche differentiation. And we are getting these terms from the competitive abilities of the two plants in the mixture. If you get a 1, it means both the plants, are using the resources within the environment to the same extent. There's no competition between the two plants. Now, if it is less than 1, it means there's a competition within the mixture. And if it is above 1, it means these plants are actually not interfering with each other within the system. But if it is less than 1, it means there could be serious competition. Now I want to stop here and switch to the PowerPoint because I thought I should explain this. So I come to the PowerPoint. Are you still there?
Yes, Doctor.
Okay. Okay, I explained these treatments. So this is what I was trying to explain to you, that if you have got density which is increasing for a plant, its yield will form this sort of a rectangular hyperbola. The yield increases up to a point, then there's no more increment. And this is how we can represent the plant competition in a monoculture. We can use this equation. where Y is the yield in a monoculture. This is density. These are just this is the slope. This is the intercept. We said if we plot an individual plant weight, this is all the plants included, we get this sort of a shape. But if we measure, if we take one single plant, we get this shape. Maybe let me explain why we get this kind of a shape. A plant growing on its own has no competitors. So we expect it to produce a lot of biomass. Once it is joined by other plants, intraspecific competition starts. Then the plants become smaller and smaller as the numbers of plants are increased. So the performance of an individual decreases as you increase the density. Now we can convert this rectangular hyperbola into a straight line by plotting the inverse of the individual plant weight, which I was representing by W. So we can get this straight line. And this is the reciprocal, the plant weight, which is 1 over W equals to this equation which we are interested in. BO plus BCD. Now, here at the University of Zimbabwe, we had actually 2 students who did some work on crop weed competition using these 2 weeds, Tithonia rotundifolia and the Erosine indica. In maize, we had the Munyaradzigata and the Tavunyakurai in these 2 different years. Now, when they plotted the individual plant weight, this was the Tithonia. No, not the individual plant weight, sorry, the biomass of Tithonia. As they increased the density, you can see there's that sort of increase in biomass, then there was no more increase. So we get this sort of a shape which describes more or less a rectangular hyperbola. Now, when they took the individual plant weights, as they increase the density, you can see this sort of a curve. And when they plotted the reciprocal weights, you can see they transformed this curve into a straight line. So this is a characteristic feature of competition. And as for Tithonia, this is the equation which can be used to express the reciprocal of the plant weight of Tithonia, which is a straight line. Here we come to the equations proposed by Spitters in 1983, where he took in a crop, in a monoculture, you could simply express the reciprocal of its weight by a straight line equation. Just like this in Tithonia. Now, this is the situation in a crop where we have a mixture. We can express the reciprocal of the plant of the crop weight with this sort of equation. This is the intercept, BCC and the BCW. These are the slopes, or just the coefficients. But we now know that BCC can give us intraspecific competition, as well as BWW here. It can also give us the value of intraspecific competition. Then BCW and BWC, they can give us interspecific competition, like what I've specified here. So all this information is coming from the addition series experiments. And its achievement is that we can now measure and separate intraspecific competition and interspecific competition, which cannot be done with the additive experiment as well as the replacement series experiment. Of course, the cost of doing it using the addition series is that you are now dealing with a very big experiment, because we have put several densities in a replacement series experiment. Now, the competitive ability of the crop and the weed can also be estimated. We can use these coefficients BCC divided by BCW to estimate the competitive ability of the crop. We can also estimate the competitive ability of the weed by dividing BWC into BWW, which we get from those equations. We can also calculate niche differentiation index, which tells us the nature of competition, how these two plants are exploiting the environment. Are they exploiting equally, or they are differing in terms of how they are exploiting the environment? And using the competitive abilities of the two, this is actually a product of the competitive ability of the crop and the wheat. That gives us the niche differentiation index. Actually, some of these terms could be applied to intercropping experiments. This is a paper by Steven Radosevich, Methods to Study Interactions Among Crops and Weeds. In this paper, he is also explaining the addition series experiments. If you get hold of this paper, maybe it may be clear. Now, this is from this paper. And these are some of the treatment combinations. I'm sure this may be easy to understand on this table. It's the same. Where A represent a crop, one plant. B also represent one plant. You can see we are varying the density of B going this direction. We are also varying the density of A going this direction. If we look at A, the proportions of A are changing, whilst those of B are constant, at 1, 4, 8, and 16. So the proportions of A are increasing in this direction. If you come again to B, you look at what is happening to A. We have got ones going this direction. All right. We have got ones in this direction, then 4s, 8, then 16. It's a special way of getting the right combinations. But these are just the proportions. So if you read this paper, you may get something useful. Now, this is a special graph which is known as the response surface. Here we've got a mixture of maize and Rapoco grass. This could be maize, dry matter. The shape of this box can give us various combinations about the dry matter of maize. We can get various combination of maize and Rapoco grass, which produce dry matter of maize. And the equations which I was showing you previously probably describe these shapes. This is known as the response surface. If you want to get further information about the response surface graphs, where they are being applied, you may have to read this paper by Regmanek et al. It's a weed science journal. It's weed crop competition, experimental designs and models for data analysis. You just get what you can. If you encounter some difficult areas, you may ignore such areas. But I think you will get something out of this paper. These are just, response surface graphs. You may see that we have got 1 over W equals to whatever these numbers. These are the sort of equations which I was trying to explain to you. But these have now been solved in an experiment. They just describe these response surface graphs. This Swanton et al. Paper, it describes everything which we have talked about, starting from additive designs, going through almost everything about these lectures. So you may find it, probably it may explain other things in a better way. This is also another paper on wheat and wild oat. It is also showing the addition series experiment. Here the title is Response Surface Experimental Designs, Investigating Inter-Specific Competition. So it's about what I was trying to explain. You may have a go on these papers and see whether you may understand what these scientists are trying to explain. But I'm sure you'll eventually get to understand these issues. Now, there's also a study which was done by Chikoe. Chikoe is a wheat scientist from Zambia, based at IITA in Nigeria. He studied the competition of Imperata cylindrica and maize using the addition series experiment. So you may read this paper and see how how this design was used to understand the competition of maize and this grass. Now, this is a paper done in Eritrea, but they are not using weeds as such in this paper. They were growing barley and wheat, but they used the addition series for their study. In fact, they wanted to find the best combinations because they grow wheat and barley in mixtures. So sometimes they try to find the best mixtures. It's like intercropping. And as I've said, this method can also be used by agronomists to find the optimum populations or combinations. You may also have a look at it, but it's not talking about weeds, but the methods are the same. Now I want to move on to talk about the dialer experiments. Now, these are less used by agronomists. And the research question may be, what is the competitive ability of two plant species? You know, weed scientists and agronomists may not be interested in individual plants. But of course, in biology, I mean, people may have different objectives. So in this case, the method is that you may have two species, A and B, which may be grown in pure stands or mixtures. Actually, the idea is that you want to measure intra-specific competition and inter-specific competition. So you can have an example of these plants. A can be grown on its own, or you can increase its population. You have two plants. Here you can have A being grown by another plant B. You can do the same with the plant B, grow it on its own, then double it. Then you can now detect intra-specific competition by simply comparing A on its own and when it is doubled, B on its own and when it is doubled. As for inter-specific competition, you can now use a combination of AB compared with the AA or AB versus BB. This is how you can separate intra-specific effects and inter-specific effects. It's the dialer approach. And as I've said, we scientists rarely use the dialer experiments. Then there is something called the neighborhood experiments. Now, with this sort of approach, you know, plants grow within the same environment, and we may want to find their impact on each other, impacts on each other. So the research question could be, what is the effect of weed position on crop yield? Now, probably if you take a field where you have planted maize, there could be weeds which are growing near the crop row and some which are growing far away from the crop row. Do these weeds have the same impacts on the maize crop? Probably this can be solved by neighborhood experiments. Now, the method which is used is that we assess weeds right, using crop as the target species. So the impact of the weeds, we have to measure the crop response when we have weeds. So we can plant weeds at various distances from the crop rows. If we have got 0 centimeters, it means we are having weeds within the crop row. Then we can have other widths at 15, 20, 30, 35, and 45 centimeters, and see what can happen to crop yield. Here I have a diagram which probably may explain what I'm trying to say. This is the crop row. We can have some weeds within this row. It could be another treatment. Then we can have weeds which are now away from this, positioned away from this crop row. Some are now 30, 35, and 45. So as you move the weeds away from the crop row, what can happen to this crop? In some papers, they talk of area of influence by plants. Plants which are neighbors starts affecting each other when their area of influence starts to coincide. Now, this is a plot of the distance away from the crop and yield. You can see as we move away from the crop, as the weed is away from the crop, yield start increasing. What it means is that those weeds which are positioned are not actually damaging the crop. So regression analysis or graphs can be used to interpret these type of experiments, neighborhood experiments. Now at the Henderson Research Station, Zimbabwe tried to use this approach. He was growing maize and rapoko grass, and rapoko grass was being positioned at various distances from the maize. So here it is 0, which means the weed was with the crop within the same row. Then he spaced the weed at 20 centimeters, then at 40 centimeters. Then he would now compare the grain yields from the maize plants. I think in the first year, 1994-95, I think there were no significant differences. You could not tell whether the weeds which were within the row and those away from the row, they appeared to have the same impact on the crop. But in this season, as the weeds were now far away from the crop, maize yields were increased. At 40 centimeters, you are reaching 5 tons. 20 centimeters, you are at 4 tons. Within the crop row, we are now achieving 3 tons, something like that. Here we've got a paper which also used the neighborhood approach. It's by Vogit and other authors. So they were also trying to measure neighborhood distance away from the target plant. Because these gray areas are representing the position of the competing plant. And this white space is where you don't have competition. Okay, you can read about this neighborhood experiment from this paper. Maybe you'll get an understanding about this experiment or this procedure. Now I'll move to the Neruda design. This is usually used in forestry, and it is used in tree density plots. We may want to test multiple tree densities just in a single plot. You know, with forestry, you may end up with very big plots because they are dealing with the trees. So when you are now having, like 1 hectare of an experiment, it becomes, difficult to manage. So an elder design can actually reduce such problems. So they have what are known as circular plots, which contain concentric rings. So in a smaller plot, you can get the information which you want about density. This will be about competition. So regression techniques can be used to analyze the data. The plots look like wheels. No wonder why they are called Neruda wheel studies. So this is an example of a plot. Now, in the outer areas, the trees are spaced at a larger distance. As we go inside, the distance is becoming smaller. So we expect more competition as we approach the center of a plot. Here we've got a mixture of two types of trees, sycamore and the ash. The sycamore has got the red, brownish squares, and the ash the black squares. As you approach the center, it's now dense. We expect smaller biomass production inside here. Then somewhere out, maybe there's an appropriate mixture which can support both the trees. Here it's the same type of plot. And these are the trees. Probably you can measure this. And the outer may be a buffer, which is not measured. Yeah, I just want to appreciate, this is the picture of these rings. You can see these trees which have been planted. Again, the nailed plot. You can see as you go inside the sender, the distances are very close. So we expect more competition towards the sender. And this competition is reduced as we move out. So when we are collecting our measurements, regression analysis may have to be used. So this is how the plots look like. This is the picture of the plots. Now, you can read this paper by Stankova. I think this is Russian, where the nail-the-wheel experimental design is applied in forestry. But for agronomists and probably weed scientists, we do not normally use this nail-the-wheel experimental designs, although they could be used in intercropping. If you want to find the mixtures to grow, very, the nailed design could be very convenient because within a smaller area, you get the information which you want. Again, this is another paper which you may read so that you can gain an understanding to the use of nailed designs. So you can see on the designs which are not very crucial to weed scientists and agronomists. I did not go into the details. So I just want to summarize what we discussed in these lectures. We started by defining weed crop interactions. For example, competition. We tried to understand what it is, adenopathy, then interference, which we said was a combination of competition and adenopathy. And also we highlighted the parasitism. Of course, later on we will talk about adenopathy and parasitism. We tried to answer the question, what do plants compete for? Things like above ground resources, like light, carbon dioxide, then below ground, nutrients and water. Then we try to define intraspecific competition. We've been talking about this intraspecific competition and the interspecific competition. Now, this is what we went through, the methods which are used to study the effects of weed density in crops. First, we looked at the additive designs. This is when we're talking about weed control thresholds, the competitive ability of the weeds expressed as the maximum yield loss and the slope. Then we talked about replacement series designs where we can assess the competitive ability of both the weed and the crop. And we said that number 1 and number 2 have got some problems which can be improved in addition series or response surface designs. They may also be called factorial designs. In fact, number 1, number 2, number 3, these are used a lot by weed scientists. Then we also talked about dialer designs. These are less used by weed scientists. Neighborhood experiments, yes, they may be used by scientists in doing agronomy or weed science. Then narrowed experiments, these we said are applicable to forestry. There are some indices of plant competition which we talked about. Aggressivity, when we were talking about a replacement series diagram, replacement series experiments. That one was actually proposed by my McGill-Christ in 1965. You can get hold of the paper if you are interested in it to see how it was, developed. The relative crowding coefficient, this was proposed by DeWitt in 1960. We also talked about it when we were talking about replacement series experiments. Cousins and O'Neill also wrote about it. Then relative yield, DeWitt, 1960. Vandenberg also in 1965. Relative yield total, it's all about, DeWitt. But of course, we now have recent work on replacement series experiments. Now, for your own information, replacement series or substitutive experiments are also commonly used in intercropping. For example, maize and cowpea or maize and groundnuts. But as for us, our focus was on weeds, but you can apply the same principles to agronomic trials in intercropping. Probably I've already mentioned it again. This paper now gives you an overview of everything which we have talked about. Everything which we talked about in these lectures can be found in this paper. Maybe on some things they are better explained. If you want to know about indices of plant competition, remember we just used about 5. probably this author is talking about over 50 indices. But at least it will give you an insight into what we were talking about. Some of the things may not be useful. Right. Some of these methods which we're talking about can actually be used to identify crop genotypes which tolerate weed infestation in agricultural systems. But I have never seen a crop variety which is said to be tolerant to weeds. But in some countries, they have developed the rice which tolerate weeds. We could have some genotypes which are tolerant to weeds, but we just don't know about those genotypes. Unless we do these kind of studies, then we'll be in a position maybe to get to know some useful varieties which may be recommended maybe to low input agricultural systems. So this is it on the plant competition or weed crop competition. I'll stop sharing. Right, so this is it about weed density. Do you have any questions?
No questions.
I wanted to talk about it's something to do with the competition. It's about information which is necessary to decide when to start weeding and how long we should weed a crop. Should I proceed now or? You want a break?
Yeah, maybe just a 5-minute break.
Then we proceed.
Yeah.
It's a short. I hope you. For me, it's a short lecture. I hope for you it may not be a very long lecture.
Okay.
Okay, let me stop recording now.